Now that we have covered populations, samples, and sampling considerations, we are ready to talk about the different types of studies a biostatistician might be designing or analyzing data from. The first type of study that we are going to talk about is experimental or empirical studies. And in these studies, researchers assign some treatments to individuals. Treatments are specific conditions that certain individuals are exposed to and others are not. For example, in a drug trial, there are individuals that are treated with a drug and others that are treated with a placebo or sugar pill. Because researchers can randomly assign individuals to treatments, a well-executed experimental study removes confounds, or variables which potentially drive an association. Your book uses the example of high-altitude mountain climbers as a confounding variable. Between 1978 and 1999, 40 out of 1,173 climbers on Mount Everest, or K2, died. Huey and Ugaska found that the use of supplemental oxygen increased mortality risk over those that did not use supplemental oxygen. One would really be tempted to say that increased mortality was associated with using supplemental oxygen. However, it might be that using oxygen was associated with being less prepared for the climb, and therefore the climber was more at risk of other factors that could contribute to mortality risk. The unpreparedness is the confounding variable here. And scientists must be very careful about which variable they compare and how they interpret their results when doing empirical studies because they're of these confounding factors. The other type of studies are observational studies, where researchers do not assign any treatments to individuals. Instead, they observe and record natural phenomenon in real time. Because researchers do not assign treatments in observational studies, observations cannot prove causation or disentangle cause and effect. Both types of studies measure variables within samples. There are many types of variables that are divided into either numeric or categorical variables. Numeric variables can be discrete, so that means they can take some values within a range and can be counted. Some examples of these variables are the number of squirrels in Central Park, the number of students in a classroom or in a school, the number of species that live in a rainforest, or the number of eggs laid by osprey within a particular year. And then there are continuous variables, and they can take any value within a given range. For example, the heights of trees are continuous. The biomass of coral produced per year is continuous. The amount of ice melt from the polar ice caps over a year is a continuous variable. The other type of variable are categorical variables. And categorical variables describe membership in some sort of group or category. For example, survival, so whether an individual is alive or dead is a category. Sex chromosome genotype, if you're XX, XY, XO, XXY, or XYY. What life stage are you at? So insect could be an egg, larvae, or adult. Size classes, small, medium, and large. Those are all categories, so categorical variables. And they can be either ordinal, which means that they can be ranked, or they're nominal, and they cannot be ranked. Size classes, of course, are ordinal, right? You can rank them from smallest to largest or largest to smallest. Tree heights are ordinal. They can be ranked from shortest to tallest or tallest to shortest. Sex chromosome genotype, on the other hand, is nominal, since there is no inherent order to the 5 different categories. Knowing what kind of variable we have determines how we analyze the data and is critical to doing statistics. In both types of studies, scientists compare variables to answer questions and to test hypotheses, we'll talk about later. To do this, they test how well one or more explanatory variables denoted as x predicts or impacts the values of response variables, which are denoted as y. In an empirical study, the explanatory variables are typically what is being manipulated in each treatment. In an observational study, environmental features like temperature, light, and nutrient content act as the explanatory variable, but are not manipulated by the researcher. There are positives and negatives to both observational and empirical studies, but both are legitimate, and the data that they produce can be utilized by biostatisticians to answer biological questions.